Hello everyone, this is Doug, and in this video we're going to talk about phospholipids, and we're going to identify what their subgroups are, what their subunits are, what their major function is, and how to spot them. So, right, it's definitely one of the classes of lipids. We talked in the first video about the triglycerides, right? That was this molecule on the very bottom here, right? So here's your triglyceride. And in this video, right, we're going to talk about the phospholipids. And so phospholipids are molecules like this one, right? They're a little more complicated than triglycerides. They look similar in that they have these long fatty acid tails and everything, but this part over here, right, is a little more complicated. So we need to talk about what the subunits are that are located in that area, and then also what are the consequences of the fact that those subunits are different than they are in the triglycerides. So first of all, the major function, right, of phospholipids is that they make up the cell membranes, right? Without phospholipids, there are no cell membranes. And so when you look at a cell, right, if someone was to just draw a cell, the idea is here, so here's my little cell, maybe I have a nucleus and I have some other organelles in there. The idea here is that if you took this little membrane, right, this little part right here, and you blew it up, this is what it would look like. So that line is actually two layers. It's not one layer, it's two layers, which is why this is called a phospholipid bilayer, right? Because there's two layers of these phospholipids. And the phospholipid is supposed to be this little thing that is this circle here. So you have a circle here, and then you have kind of these two little tails coming off of it. That's supposed to be one phospholipid here. So that would be your phospholipid, that little thing. You notice that there's a whole bunch of them and they line up a certain way. So this would be the phospholipid head. And then you'd have your tail here. And we'll talk more about the hydrophilic head and the hydrophobic tail in a second, but yeah, this tail is hydrophobic, it's not attracted to water, and the head is hydrophilic. And so the idea here is that these all line up so that, right, if we have water on the outside and the inside of these cells, sorry, the outside's the top part, the extracellular, then the intracellular, right, is the inside of the cell. We have water here, so the hydrophilic parts of these molecules are attracted to water, and the hydrophobic parts sort of hide away right here, away from water. And what's cool about these phospholipids is if you just take phospholipids and you just chuck them into water, they automatically form micelles, which we can talk about later, but they also form these bilayers just automatically. So these cells just kind of just form, these membranes just sort of form. And then, of course, more complicated cells, right, have these differentiated organelles like mitochondria and a nucleus and all those sorts of things in there. So what I want to do is take a little closer look at how we relate this little round sphere with two yellow tails to the actual subunits, which are glycerol, two fatty acids, a phosphate group, and a polar alcohol group. So let's take a look at these subunits a little more closely, right? We've already mentioned glycerol, so we already know what glycerol is, right? So glycerol is kind of the backbone of phospholipids, just like it was the backbone of fatty acids, right? So here's your glycerol piece again. So we've got our glycerol piece, that's the same. Ditto our two fatty acids, right? That's the same also. So again, here's my fatty acids, right? So I have this long fatty acid, I have this long fatty acid. And again, just like with the triglycerides, these fatty acids can be trans fats, cis fats, they can be monounsaturated, polyunsaturated, etc. Remember that trans fats are real bad, so hopefully you don't have a lot of trans fats going on here. What's different about the phospholipids as compared to the triglycerides is the fact that phospholipids, as the name indicates, right, have phosphates. So here we actually have our phosphate. So phosphate group. Phosphate, phospholipids, makes sense. And then lastly, the kind of like the wild card, the thing that can be a lot of different things, is this what's called the polar alcohol group, right? So by alcohol, we mean that it has to have a hydroxyl group, so it needs to have an OH. And polar just meaning that it needs to have like full charges or lots of partial charges or something like that. So it's gonna be just very polar is the idea. It's gonna really, really attract water. And in this case, I drew this molecule right here, which is gonna be my polar alcohol group. In my case, I have choline. You may have heard of phosphatidylcholine. Choline is important for brain function, and it's in phospholipids, etc. And again, just like with the other macromolecule subunit relationships, right, if I want to combine these two, right, so here's all of my subunits up here. If I want to start combining them, right, I need to start circling off my water molecules in order to get my phospholipid, right? So in this case, I'm going to do 4 condensations. So condensation. That's going to give me 4 water molecules. And then my actual phospholipid, right? So here's my phospholipid down here. And when I combine all those things, right, the idea here is if I combine all of those different pieces, right, so I've got my two fatty acids, they're attached to glycerol now, I've got my phosphate and my polar group. I'll even highlight those right here. So here's my polar amino group, here is my phosphate piece, here are my fatty acids, right, that are still attached, and then lastly, here is my glycerol piece, right? So I've combined all of those together to get my big macromolecule. Now, if you take a look at this, right, you can break this into 2 pieces. If you take a look at this piece over here, all of this stuff right here, right, look, I've got a full positive charge, I've got a negative charge, I've got lots of partial charges here, right? That'd be partial, negative, positives, negatives, negative, positive. I've got a whole bunch of partial charges over here on this end of the molecule, and I don't have any partial charges on the right-hand side of the molecule. So if I'm asking myself, well, which piece of this molecule is the more polar one, or in other words, the more hydrophilic one, right, it looks like this piece over here is the hydrophilic piece. So this would be hydrophilic, right, because it's polar, right, that means, right, it's going to attract water. Right, this piece over here is going to attract water. So this is your hydrophilic head. So that was that red circle that I drew on the previous part, right? This would be the head of your phospholipid. Versus, right, we said that if I had, if I take a look at this piece over here, so all of this piece and this piece, right, these pieces are hydrophobic because they're just nonpolar, right? So these pieces over here, right, this is all nonpolar stuff. Right, I just have carbon-carbon, carbon-hydrogen. That means it's hydrophobic. And that's what makes our tails, right? These would be our tails. Right, so those 2 yellow tails that I drew on that previous slide, these are the 2 tails of that phospholipid, right? So again, like in that previous slide, I drew the phospholipid head, and then it had 2 tails. That's what we're talking about, right? So we have that really hydrophilic polar head that's going to be attracted to water. It's going to be on the outside or the inside of the membrane. And then we have the hydrophobic tails that are going to line up against each other so that they stay away from water, is the idea. All right, so they stay away from water. And that's kind of what a phospholipid looks like. That's the basis of a phospholipid. How can it change? The fatty acids could be different, right? In my case, I have a trans fat, which is not good. Hopefully you don't see any of those in real phospholipids because you don't want to eat them. But it's a trans fat. This top one up here, right? This one right here is a trans fat, and it is polyunsaturated. This one down here is a cis fat, and it's also polyunsaturated. But there's no reason why they can't be saturated or they can't be monounsaturated. It's just kind of whatever fatty acids you're eating in your diet plus the ones that your body's making. So it'll be some mixture of whatever. So those could be different. Phosphate will always be phosphate. Glycerol will always be glycerol. But these polar alcohol groups is kind of a big group, right? They can be a lot of different things. So I want to show you some examples on the next slide here of things that they could be. I did choline for mine, but it certainly doesn't have to be choline. There's other things that make other kinds of phospholipids. So like for example here, right? So we're saying that the polar alcohol group can be different things. I did this one up here, right, where I had my polar my polar alcohol group was choline. So that's the one I did in the previous slide. But you'll notice that here's one that's a lot like it. This is, oh, there's one too many carbons here. Sorry, forgive me. I don't know if anyone's counting. One too many carbons, so ignore one of those carbons. But these would be polar alcohol groups, right? Because this used to be NH3 plus. This is supposed to be ethanolamine, not propanolamine. It's supposed to be from that one, so just 2 carbons here, not 3. But again, it's an alcohol group and it's polar. If you take a look at this group down here, right, There was an OH here, that's how it did the condensation, but certainly very, very polar, right? We have a whole bunch of partially positive and negatives, negatively charged atoms here, right? It looks kind of like a sugar, even though it's not, and just in that it has a lot of alcohol groups. And then we can also have things like amino acids, like here's serine that got put into a phospholipid. But again, super polar. It's got lots of partial charge. It's got charges on it and partial charges on it. But these are just different groups, right? These are just different polar alcohol groups that you could find on a phospholipid. The rest of it's the same, right? We have a phosphate, we have glycerol, and then we have 2 fatty acids. I didn't draw the whole fatty acids. I just put an R group here. But the idea is that polar alcohol group can be lots of different things. So don't freak out if you see something there that's not familiar to you. As long as it's pretty polar, it's still going to be a phospholipid. And so just to give you kind of an idea, I'll come back to this when we talk about steroids too, but right here's another view of what these phospholipids look like here, right? So if you get a whole bunch of these phospholipids, you end up making this phospholipid bilayer, and then you can start to see how other things fit into that phospholipid bilayer, right? So here's just like a cutaway section. You'll find things like these proteins, right? There's channel proteins, like right here, things that go all the way through that allow things in and out of the membrane. You have different proteins that can kind of go through and then help signal. We've mentioned before, right, that there are glycoproteins and glycolipids. These are sugars on the outside of your cell that are signaling whether it's part of your blood type or whether it's so that your immune system doesn't attack, right? That's a way to have some cell recognition. There are other kinds of proteins here. There are some that just sit here and do different things. You guys will learn more about proteins that are embedded in cell membranes, like in the electron transport chain, for example. There's all kinds of things. The phospholipid bilayer gets more complicated. And then in the next video that we're going to talk about are things like cholesterol. You'll notice that cholesterol is in your cell membrane also, and it's really, really good and really, really important. So we'll talk about that in our next and last video. So I want you to have so hopefully you have a better idea of what a phospholipid is, what its subunits are, right? What the pieces are that make it up, and then how important it is because they make up the cell membranes. Without cell membranes, you don't have cells and you don't have life as we know it. So we'll come back and we'll talk a little bit more about the phospholipid bilayer when we talk about steroids. If you have any questions, please go ahead and leave them down below. Happy studying.